Hello,
Hello.
how are you?
Yeah, I'm fine. How are you?
Pretty good, pretty good. Welcome to the meeting. Hello, Jesse. So, Himanshu, did you want to give your update?
Yeah, just a second.
Um, Sushmanth won't be joining us today. He met with us Friday. He has exams, so
Oh, okay.
yeah.
Is my screen visible?
Yes, can see it.
Okay, so my update of this week is like I've been working on the model that I proposed last week, the mass car CNN model. So one of the things that I was doing is the creating the bounding boxes for all the cells that are present like in the data set. So I worked on that as well as I tried to do it in some different way, like do it without using a function and doing it on my own to see how I can make a better way of getting the bounding boxes. And other than that, I've been working with this data set, which was always also used in the previous year GSoC, as well as the new data set which I had shown last week. Last week, the data set which I mentioned, the data set that was just a second. Yeah. So it's this one, the one with the L1 stage of C elegans data, which is also uh, shown in this paper. What I also inferenced through this was like I researched more about the data set, and basically the data is about the first larvae stage L1 of S3 elegans, and they have used a confocal image stack of 15 different individuals, as well as they have inference 3 and segmented and allocated 558 develop 558 annotations at the developmental stage out of the 357 nuclei that are present in the worm. And they have also done like annotations manually as well as used a computational method on the dataset as well. So after completing the model, I'll be running it on this as well. And I've also been reading about temporal data analysis and getting up to touch with that as well, because I'd be working on it the next few weeks. Like the one paper which you had also given in the Slack channel, the paper which was referenced by Jiahang as well, about the temporal data analysis which was done on C elegans. And I've also read this tracking everything and everywhere all at once, which was like something which Sushmanth is also referring. And yeah, that's my update.
Oh, okay, that's good. So yeah, you're working on the data sets. You're getting established for starting data analysis once you get the segmentation part done, and then you're on your way. You're doing yeah, I think that's good. Any roadblocks coming up? Do you think, or do you think you're on the right track as far as you can tell?
As far as I can tell, I think I'm on the right track right now.
Okay.
If some problem arises, I'll DM the mentors, or we have a group as well on the Slack, so I'll write that over there.
Okay. What is your timeline for sort of getting to the analysis stage?
I will be doing it this week. Like
Okay.
I'll be finishing with the segmentation, instant segmentation part until this week, and hopefully show you it in the next week. So that was the original timeline, and let's see. I'll try my best. As well as then, and the way I had given the timeline was like the first 4 weeks for segmentation, the next 4 weeks for the Devo graph part, like to derive better graph embeddings, and the next 4 weeks out of after that was the final part, which is to combine DevoLearn and DevoGraph into a combined, like using DevoGraph as a submodule for DevoLearn. Yeah.
Okay. That's good.
Yeah.
So yeah, what kind of tool? You're just using Python toolboxes for this, or are you using a specific tool? Especially when you get into the data analysis stage. What kind
Right now, I'm just using Torch Vision and all the features it gives us. So, like for bounding boxes, like we have a tutorial as well for like a util function for it, and otherwise as well, I'm using like I've been using Torch Vision on. Yeah. Also, like PIL and other libraries for like image, looking at the image and so on, whichever is the most efficient way to do it.
Yeah. Well, that looks good. Yeah, thank you for the update. And you're finding those data useful, the data from the larval stages.
Yeah.
Yeah. So yeah, that's really good. Thank you for that. Now I got a message from.
Oh.
Oops. You just stopped sharing your screen, I guess.
Yeah.
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 There you go. So yeah, I got a message from Jiahang this week, and he was interested in doing a paper on some of the things he did last summer and some of the things you're doing this summer. And I said, yeah, it'd be great. So I don't know if he's gotten in touch with you about that, but he's interested in doing and this is a little bit longer term than, in the next few weeks. So we'll be working on that, hopefully. And I just messaged him earlier, and he said he might be here a little bit later. He's running late. So yeah, are you interested in doing that sort of being involved in a paper like that?
Yeah, I'm definitely interested. Also, like graph neural nets is something which I wanted to get into more and also research about it as well. So
Yeah,
so yeah, I'll be interested.
yeah.
Also, Jan didn't mention it this yet. I guess he would be mentioning it this meet or something.
What was that?
Jan didn't DM us yet, like me and Sushmanth. So probably he'll be mentioning us this week or
Oh
something.
yeah. I don't know. We didn't get around to it, so yeah. But yeah, we'll be talking. I don't know. Maybe hopefully it comes in a little bit later. We can talk about it, but if not, we'll just talk about it in the Slack. Um, yeah,
Sounds good.
yeah, good. Thank you.
Thank you.
All right, so yeah, like I said, Sushmanth is out. He has his exams. He's doing well. He gave an update, and we have a Friday meeting on open source, and that's usually for the Orthogonal Lab group, which is a different group. They're doing different projects. We usually meet then and talk about like their projects, and I usually give some updates on like open source topics. And so that's actually on YouTube. You can look it up on the Orthogonal Research and Education Laboratory YouTube channel. So I mean that's something. If you feel free to attend if you want, but we'll be doing your main updates in this meeting for the DevoWorm people. So just to clarify that. Like I said, Jiahang said he might be able to make it later in the meeting, so we'll reserve some time for him to do that. Let's see. I'm going to share my screen now. And I'll give a talk on a few topics here. So I think I mentioned this in the meeting, in the Saturday meeting for the orthogonal lab, that I was going to go over some papers on making human embryos, which is, there have been a couple papers that have come out recently on this. I'm going to go over the papers with the understanding that I'm not I don't know a lot about this. I mean, I know about embryos and I know about organoids and, reprogramming cells, but, this is definitely something that is, okay. So Himanshu said, I'll try to attend Friday's meeting. Yeah, it'd be great. But it's definitely sort of a cutting edge area. So this was posted. We have a channel in our orthogonal lab Slack, which is a brain organoids channel, and so we posted these in there. This is where we monitor a lot of the organoid work as well. So this first paper is called Transgene-Free Ex Utero Derivation of a Human Post-Implantation Embryo Model, solely from genetically unmodified naive PSCs, or like stem cells, basically. We'll get into what that is in a little bit. This is a number of authors, Jacob Hanna, who's done a lot of work on embryos and implantation in the past. So this is the first paper. The abstract reads, our ability to study early human post-implantation development remains highly limited due to the ethical and technical challenges associated with this kind of development. So the difference between C. elegans and humans, of course, is that C. elegans develops in an egg, and humans develop in an amniotic sac, or what is an amniotic sac evolutionarily. There's this implantation stage where it gets implanted in the uterus, and that is key to life-success of that fertilized embryo making it to a live-born human. So this is a stage where there's a lot of work that's been done on what they call indirect reprogramming, which is where they take a somatic cell and they put in a nucleus or they take a stem cell or a germ cell and they put in the nucleus of a somatic cell. So this is the egg or the ovum, and then it has a nucleus. They take that nucleus out. Then they take a somatic cell from an adult. They put that in a gamete, and then that now has a genome that's fully adult, but it's in a germ cell, so it's reprogramming the cell. That gets implanted into a uterus, and it grows to a clone, basically. So this is the human cloning work that was done probably about 25 years ago now, first done, and they've been able to do it ever since. If you think about something like cellular reprogramming, which is where you take a cell, like maybe a skin cell, so the same type of differentiated cell, and you introduce some viral vector with some factors. So there are transcription factors in this, which just means that they're genes that need to be expressed for the cell to change, maybe change its fate or change its function in some way. A viral vector is just like a transgene that you can put in through a retrovirus, and it just infects the cell and it goes into the nucleus and it inserts itself in the genome. And then from that you get basically this stem cell. It's not really a stem cell like we might find in an embryo, but it's a stem cell. It basically expresses all the markers of a stem cell. So that's direct reprogramming. So this is indirect which means that it's kind of you're putting the you're putting the genome in the cell, and it's not integrating, it's just turning on some genes. Direct means that you're putting genes in the genome of a cell and you're sort of directly programming its function. So that's the distinction. And so, we have human development, we have something like C. elegans. C. elegans, of course, has an egg where you have cells that divide. 2-cell stage, 4-cell stage, 8-cell stage, and that. In humans, you get this embryo that's like this. You get maybe up to like a blastocyst, and then, which is a ball of cells. And it starts to differentiate into something with an inner cell mass. This is true of a lot of mammalian cells, or a lot of mammalian embryos. And then this embryo has to implant itself in the uterus of the mother. And that successful implantation leads to a production of a viable embryo that becomes a young organism, which is not a clone. So this is what they're talking about here. And they're talking about this implantation step, and then post-implantation, which is where it's actually developing in the context of basically it's being supported by the mother. So there's a lot of biology here that I'm not going to get into. So it's important to recapitulate this step. So what we can do now, of course, is we can create organoids. Of course, we've talked about organoids, and it could be something like they have these weird shapes because they're not really. They have some directional cues, but they're still kind of this mass of cells. But they have layers, and the layers are, of course, different cell types that are maybe functionally associated, maybe not, depending on how good of a job you do with the organoid and raising it in this 3-dimensional cell culture. You can get cell signaling between different cell types, and so that's that's the organoid work. Now this is actually creating embryos, and so they're talking about some of the work that they're doing here. So they're able to sort of, they need to recapitulate this early cellular environment. They need to recapitulate this reproductive environment. And so how did they do that? That's been the big stumbling block to, being able to basically clone organisms. So they can do this with like sheep and goats and cows and things like that. They've been able to do indirect reprogramming. But a lot of this is, hard to do, especially in humans. And so you need to be able to recapitulate post-implantation epiblast development within the context of these extraembryonic compartments. Mouse-naive pluripotent stem cells, or PSCs, so these are pluripotent stem cells. And we've talked about pluripotency before, which means that you have a stem cell that can become multiple types of mature cell or adult cell. So you have maybe a pluripotent stem cell that becomes different types of neural cell, or different types of, just different types of cells. It can't make any type of cell, but it's restricted in its set of fates, just a little bit. And so that's, this is the type of cell they're talking about here. So they're talking, in this case, about mouse and not human. But mouse-naive pluripotent stem cells have recently been shown to give rise to embryonic and extraembryonic stem cells capable of self-assembling into postgastrulation, which is a stage in development, mouse SEMs, while bypassing, and SEM is this stem cell-derived embryo model, while bypassing the blastocyst stage and eventually initiating organogenesis ex utero. So this means that they are able to have these pluripotent stem cells. They give rise to different types of embryonic and exembryonic stem cells. So they transition to another type of stem cell, and they're able to self-assemble into these different models that this type of model that they're trying to build, it bypasses some of the traditional stages of development and initiates organogenesis, meaning that you can build organs in one of these stem cell-derived embryo models. Here we implement critical adaptations to extend these findings to humans while using only genetically unmodified human naive PSCs, thus circumventing the need for ectopic expression of lineage-promoting transgenes. Such integrated human SEMs recapitulate the organization of all known compartments of early post-implantation stage human embryos. So what that means is that you have the mouse model that they've been able to do successfully. And they've been able to kind of get around this step of implantation and they've been able to sort of fast forward through development a bit and get some of these structures that we're interested in. So this is different than your typical organoid in that the organoid isn't, it can generate some of the cell types of organs, but it doesn't necessarily, recapitulate some of the compartments that we need for early post-implantation stage human embryos. So some of these include epiblast, hypoblast, extraembryonic mesoderm, and trophoblast surrounding the latter layers. So you can see these layers of tissue in the form. You start to get the formation of tissues in these type of models. The organized human SEMs recapitulate key hallmarks of post-implantation stage embryogenesis up to 13 to 14 days post-fertilization, such as bilaminar disc formation, epiblast luminogenesis, angiogenesis. So these are different, like amniogenesis, which is the genesis of an amniotic sac, bilaminar disc formation, anterior-posterior symmetry breaking. So this is where you get differences between the front and the back end of the embryo, primary and secondary yolk sac formation, and extraembryonic mesoderm expansion that defines a cavity and a connective stalk, a chorionic cavity. So these are all things that you see in embryos post-implantation. They lead to different structures in the in utero form, and so they can basically they can build a model of an embryo, and they can fast forward past implantation, and they can get sort of the origins of some of these structures. And this is, of course, useful because you can do this in a lab. You can look at, some of these structures, how they form, and you can modify them as needed. So this is a sort of, I don't know if it's really an ethically gray area. It's not like we're trying to actually clone humans, but we're like trying to sort of recapitulate some stages of early human embryogenesis to see if we can form tissues and we can do other things like that. So this new platform constitutes a tractable stem cell-based model for experimentally interrogating previously inaccessible windows of human peri and early post-implantation development. So they really kind of gotten to the point where, we can actually, organoids are these 3D cell cultures where you're starting to get differentiated tissues. You're starting to get, these sort of tissues living in proximity. This is maybe one step beyond organoids, where we have some of these features of early development. And so this is, they've done some of this in mouse. Now they're doing it with human embryos. And so there are a lot of genes that you need to express in some of these cells to get them to move to certain fates. And so they're able to do that. Some of that can be done chemically or through a transgene or something like that. But you can also do it just by having tissues in association with one another and getting cells to differentiate and form like layers or structures or compartments, as they call them. So the second paper is the stem cell-derived mouse embryos develop within an extraembryonic yolk sac to form anterior brain regions and a beating heart. So this is about mouse embryos. This is if we back up and we say they're mouse embryos that we can develop, or these mouse models stem cell derived embryonic models. We can do this in mouse. Again, mouse development is very similar to human development in the sense that there's this implantation step. It develops in an amniotic sac. It develops in a uterus, and that step, of course, is also necessary. So mouse embryos develop within an extraembryonic yolk sac to form anterior brain regions and a beating heart. So we're able to recapitulate this ability to embed this thing in a extraembryonic yolk sac. So this is extraembryonic. This is outside the embryo. You have a yolk sac. You're able to form these different structures. So in this case, you're forming anterior brain regions, or things at the front of the brain, like the forebrain, and then a beating heart. So it's not only you're forming heart cells that differentiate, you're forming this structure that starts to beat, and you can get a viable organ in that way. So you get organogenesis, basically, in this kind of model. So the abstract reads, so this is the authors here, Magdalena Zernica Getz, who is a famous person in this field, and a couple other people here. So the abstract reads, embryo-like structures generated from stem cells can achieve varying developmental milestones, but none have been shown to progress through gastrulation, neurulation, and organogenesis. So these are, again, stages of development that we expect in an embryo, in an implanted embryo. Here we show that ETIX mouse embryos. The ETIX is I don't know if they get into the definition of this, probably in the paper. Established from embryonic stem cells aggregated with trophoblast stem cells and inducible extraembryonic endoderm stem cells. So these are mouse embryo models that are established from different types of stem cells. So they're just putting all these stem cells in association with one another, and they're seeing what happens. This aggregate can develop through gastrulation and beyond. So you can take that aggregate through different developmental stages. And you can do this to undertake neural induction, which is a stage where you get the induction of neurals, like the neural tube and the neural crest, and generate the progenitors needed to create the entire organism. So you can get this sort of neural differentiation, and from that you get progenitors that you need to create things like the forebrain of the forebrain. The head folds of the ETIX embryos show anterior expression of OTX2, defining forebrain and midbrain regions that resemble those in the natural mouse embryo. ETIX embryos also develop beating hearts, trunk structures comprising a neural tube and somites, tail buds containing neuromesodermal progenitors and primordial germ cells, and gut tubes derived from definitive endoderm. A fraction of ETIX embryos show neural tube abnormalities. So what that means is that you get these ETIX models, you get head folds which show an anterior expression of this gene that we know is a marker for neural development. This defines the forebrain and midbrain regions, so you get this distinct region formation. So there's these differences that you can identify in these models. This looks like natural mouse embryo if you compare them. Then they also develop beating hearts, trunk structures, which is the sort of the formation of a neural tube. So you get these areas that are sort of analogous to the mouse embryo where you're getting neural development. You're also getting the neural tube, which is where you get this tube of cells that kind of form what's going to be the nervous system and the spinal cord and things like that. So you get this long tube that forms in the embryo. And so you're starting to get that as well. You're getting somites, a tail bud containing neuromesodermal progenitors. So you're getting these tail bud, so it's like the tail of the mouse, and primordial germ cells, which are different types of germ cells that you'll need for different types of stem cells. So that's all there. Then a fraction of ETIX embryos show neural tube abnormalities. So a fraction of these kind of embryos that are being generated actually show different abnormalities. So they're not all perfect. They don't all come out the same. Sometimes they come out with errors or abnormalities. What's interesting is you can partially rescue these by treatment with a metabolically active form of folic acid. So this actually resembles common birth defect therapies in humans. So they do this with birth defects. They treat the embryo with folic acid and they can maybe reverse or arrest some of these birth defects. Normally, ETIX embryos also develop a yolk sac with blood islands, so they actually start to develop blood and a circulatory system, perhaps. We're not really quite there with this, and with as well as with the yolk sac, but it's you're getting the precursors of that. Overall, ETIX embryos uniquely recapitulate natural embryos, developing further than any other stem cell derived model through multiple post-implantation stages and within extraembryonic membranes. So again, we get this model. This is in mouse, not in human, but we can do this in mouse. We can sort of see that you can move beyond like organoids. You can move beyond even, other types of model where you have these complex tissues that form. So it's a really interesting sort of set of advances towards this goal of like maybe creating creating an artificial embryo or creating an artificial human or mouse. And again, this is basically just taking stem cells. You're not reprogramming them necessarily, like I described before. You're taking these stem cells and you're putting them in association, and you're doing some other things to elicit some of these changes. But what you get at the end is this highly structured, system that actually resembles development. It isn't development itself, but it resembles development, which is important to remember because you get, there are a lot of ways you can assess what you have, and it may not actually be developing embryo. It might be something that resembles it, but it may not be something we could actually take to full term, as it were. So ESC's embryonic stem cells, which are derived from the epiblast, show a remarkable ability to form embryo-like structures upon aggregation. So we have ESC aggregates called gastroloids, which if you embed them in matrigel, which is this medium that they often use to do 3D cell culture, can form trunk-like structures with somites, a neural tube, and a gut. So these are all like things that we're looking for in the embryo that you can actually recapitulate in gastroloid models, which are different than this, what we're talking about here. They're a bit different than embryoids because they have this they're at the stage of gastrulation and development, which is where you start to see some of the structure. Although anterior neural development can be promoted in gastroloids by inhibiting the initial burst of Wnt activity, which is a gene that gets expressed early in development, and it's important for developmental signaling. They do not faithfully represent the anatomy of natural embryos. So embryoids generated from naive ESCs are these embryonic stem cells aggregated with an ectopic morphogen signaling center, which is where you have this chemical signaling which promotes morphogenesis, can develop a posterior midbrain, neural tube, cardiac tissue, and the gut tube. So you can actually do this in these gastroloid models. You can build or embryoid models, which you can actually go beyond the gastroloid model to develop some of these tissues. These models form some epiblast structures de novo, meaning that they do this within the model. You don't need to really induce them. But do not, unlike natural epiblasts, they do not undergo gastrulation. So you have these different types of models. You have the stem cell. We have stem cells, of course, which are naturally occurring. We can harvest those from different biological samples. We have induced stem cells, which are ISCs. And this just means that we can induce these with, say, like a transgene. And we just, we figure out which genes are needed to be expressed to get like a cell to the stem cell state, which means that they can produce other types of cells, precursor cells and mature cells. Then we have these organoids, which we can use to study organ formation, but they don't necessarily give us a window into development. They just give us a window into this, differentiated state. Then we have gastroloids, which allow us to look at gastrulation in very early development and the formation of some of these organs or organ systems, at least as they're forming. So in organoids, you tend to get a lot of tissues that kind of are together in the same structure. In gastroloids, you're kind of recapitulating gastrulation. And then in embryoids, you have things that resemble an embryo, so you start to get, a beating heart, you get this, you get blood clots, things like that. And then we get these models that we're talking about today. So this is this model here. So this is the ETIX model. And the model that we talked about in the other paper, which was this, let's see, SEM. So, these acronyms are somewhat kind of all blend together at some point. Okay, let me start over. So the SEM, which is this model of, another model of embryogenesis. And so these are relevant to mammalian systems like mouse and human. Right. So this is different from C. elegans by far. This is, describing sort of this complex embryo, or this complex embryo, more complex embryogenesis you'll see in what we call regulative development. You'll have, these different stages that you don't see in C. elegans. C. elegans is just kind of, the cells differentiate, they form this set of organs and tissues, and then the egg hatches, and you get this, you get these different larval stages that allow you to have a little bit more differentiation and new cells that are sort of useful in the local environment, and then you get the adult stage. In mammalian embryos, it's far more complex, and so this is these are some of the most recent forays into this, and there's been some criticism of this online. So, thinking about this in terms of human embryos and like creating a human embryo, we're nowhere near that. As I said, you can create tissues in association, but that doesn't mean that it's a human embryo. And so some of this is where you characterize the cells using things like next-gen sequencing. You look at RNA expression, you look at gene expression, and you look at like the marker RNAs that need to be there, and that's fine, but that doesn't mean that those cells are identical to what you might find in an embryo. So for example, with our induced pluripotent cells, we think that they behave like pluripotent cells or like stem cells, but they aren't necessarily those kind of cells. A lot of times, induced neural cells are like that, where you can create a neuron that has some sort of action potential, but it isn't like any kind of neuron you see in nature or in the human body or any other organism. So these cells, these are kind of like facsimiles where we try to induce the right factors and we maybe get something functionally similar to what we want, but it isn't that thing. And so this is what A. Martinez Arias talks about in this post that he had on it was another paper that I'm not talking about here, but this is a very similar problem. So beside from all of those identification issues and identity issues, to put things in perspective, a human embryo at stage 14, which is the stage of mammalian development, There's like mouse 14, but human embryos also have a stage 14. It's not completely analogous, but basically it's at this stage of development. A human embryo at stage 14 is a complex structure shown below with yolk sac attached to the epiblast that will form the embryo, and both covered by two membranes, the chorion and the trophoblast. So you see here that there's this amnion, the amniotic cavity, the epiblast is here, the hypoblast is here, and then you have this chorionic cavity. So you have all these structures that are, you have the degenerating primary yolk sac, maternal blood vessels starting to innervate the structure. So you can see all these things that are there that they also have to be well timed. So if they're out of sequence, you don't get, so this is 14 days post-fertilization, you don't get a viable embryo. And so there are a lot of things that can go wrong here. And so this is what you see in an implantation, implanted embryo, and its associated function. So we're definitely not there yet. We can get differentiated tissues, we can get other structures, but we can't necessarily. This isn't like cloning humans. Is my takeaway message from this. But it's interesting stuff. And especially in the light of a lot of stuff we've talked about with differentiation and other types of systems like organoids and things like that. So that's all I'm going to say about that. I see Jiahang's here. Welcome.
Oh, hi.
Hi, how are you?
Yeah, I'm fine. You?
Yeah, I'm good. I'm good. Yeah.
Thanks. Yeah, I'm a little bit late today. Because I was busy at something else, and I'm just got home a few minutes ago, I think.
Good.
Yeah.
So you had mentioned that you wanted to work on a paper, and this would be on, I guess, graph neural networks in the topological data analysis area. And so Himanshu's here, who of course is doing the Summer of Code for that. So did you want to talk about that a little bit?
Yes. Yes. Actually, I want to share one idea regarding the topological data analysis. Actually, the persistent homology specifically, and the graph neural network, and how they can be applied to the cell tracking. And for now, I choose cell tracking as a downstream task because I think I'm a little familiar with cell tracking, and it seems that many deep learning works have been successfully applied to cell tracking, but maybe there are some other choices, but I'm not very sure. But I wanna share the idea here.
Okay.
And wait a moment, I wanna share my screen. Right. Can you see my screen share?
Yes.
I share one paper, and actually, this paper, the graph neural network for cell tracking in microscopy videos. Actually, I think we have discussed this paper maybe the last year, and GSoC 2022, and maybe you still remember that. And this paper is actually the starting point of my idea. And for tonight I wanna briefly introduce some papers. I won't go into their details, but I would like to describe my motivations. So first of all, this paper, I won't introduce the model design of this paper, but I would like to introduce some related words that's introduced by this paper. For example, cell tracking is a very prevalent, is a very important task, and essentially in this task you want to build some cell strategies which connects cells across adjacent frames. For example, you have a microscopy video, and you want to sample some frames, frame by frame, and for each frame you have a lot of cells, but you only have the pixels or the image patches of the cells. And at first, you want to use some cell segmentation model, such as some CNN models, ResNet, BGG, or something else, to segment the patches of the cells, so that you have one patch for each cells, and each patch will contain some pixels. And with these patches, you can use some CNN models to generate the embedding of each cell based on the segmented patches. And then we go to the cell connection task. I'm sorry that I didn't. I just show the pipeline.
Yeah. Okay.
Yeah. Yes, and this and for now you have an input. You have a video, and you sample some frames, and then for each cell you will have a patch. You will have a patch of pixels. Then in the feature extraction stage, let me zoom in. In the feature extraction stage, you will have some kind of CNN models to like map each patch into the embedding of the cell. And there are a lot of CNN models you can choose, but it is not very important in my presentation because this is not my focus. And so that you will have an initial embedding, like this is the CNN and some other complicated modules. And then after that, actually you will have a points cloud, temporal points cloud. I mean, for example, for each frame you have a points cloud, and actually this point cloud will change along the time. Yes, so you have a way to convert this temporal points cloud into a directed graph. And I'm not sure if if in this paper there is any picture can illustrate my idea over the moment. Maybe not. So let's go back to the same picture. Yeah. So it's very easy. It's a very intuitive way to build a directed graph. For example, for a frame, you have a lot of cells. You can use something like KN graph to connect the cells which are very close to each other. And also you can connect cells between adjacent frames if you think these cells they are actually very close to each other in a spatial dimension. Yeah. So that means you have a way to build a directed graph. And next, you want to connect cells to build the cell trajectories. This is what we do in the cell tracking task. And it can be done by a very, how can I say, very classical task in the graph neural network, which is the link prediction. That means when you build a graph, you want to predict which each are the real edge. They are existing. You want to predict this stuff, and if there are some edges which you predict are not existing, you will remove this edge, and the remaining edges are the edges that connect cells across frames, so that you can build a cell trajectories because these edges they connect cells, frames by frames by frames by frames, and something like this. And by now do you have any question? Like, yes.
I don't think so. Himanshu, do you have any questions?
No.
Okay.
Okay. Actually, you can interrupt me anytime during this presentation. Actually.
Okay. Yeah.
Yeah. Actually, you will find that in this paper, the authors propose a way to construct a graph structure, or we can say a network structure, on the microscopic video. based on the microscopic video. And actually, there are many other papers also adopt this idea. Like we look at the cell development process from the perspective of a network, of graph, of points cloud, or something else. So when you have a points cloud or when you have a network structure, you can investigate the geometry, topology, and something else from this points cloud. This one very classical story in machine learning and deep learning. Like when you have a data sets, actually you have a set of points. But many papers would assume that there is an underlying manifold underlying this points cloud underlying these data sets, and you need to adopt some ways, some methods to explore the geometry of this underlying manifold, and this geometry and this topology will be very helpful for the downstream task. So the story also applies to here, and so the starting point is, if we have a method, or we can say we have a perspective to build the whole cell development process as a network, how can we explore the topology and geometry of this process? And actually, another question is it really reasonable to explore the topology? Because what I mean is, if we want to explore the topology of these data sets, we need to have some clues or some evidence to show that these data sets, they have some significant or important topological features. If not, to explore the topology of these data sets is not reasonable. In my opinion, I think. So here are some papers. Actually, here are some papers, and some of them have been covered in our meetings before. And there's one, Reddy's paper, I think, self-differentiation process as spatial network. And actually, I have read this paper in the last year in GSoC 2022, and actually in this paper the authors, Reddy and Richard, authors have proposed have utilized many network analysis techniques such as a k-means cluster analysis, and such as clique analysis and community detection and modularity. These techniques to analyze the network structure, and there are a lot of results, and I am not able to cover them today because I don't want to take up too much time. But the thing is, all these results will convey some interesting conclusions. For example, the connectivity is a very important property in the cell development process, in the network structure within the cell development process. And for example, if you look at the conclusion, you'll find we can see that cells in C. elegans embryos only partially segregate by lineage, sublineage, and cells that are co-located may serve as so-called weak connectors in the network representation. So in my understanding, I'm not sure if I understand it correctly. In my understanding, that means in a certain sublineage, those cells will be connect to each other very closely. But between the sublineage, these cells, the connections between these the cells across different sublineages are very sparse. So that means with the information of connectivity, it will be very helpful for us to analyze if these two cells come from the same sublinear tree, or they come from a different sublinear tree, and some related information like this.
I think you have that about right. And like the things, some of the things you mentioned in this paper were like, you have these spatial networks. So the networks are usually network theory. They're doing networks on, they create networks from data. So it could be like social media networks, or it could be like communication networks. And the networks aren't necessarily spatially defined. So you just say there are two things that are associated. It could be like that they're correlated, or it could be that they're just kind of like in proximity in time, So they don't have a distinct spatial location. In the embryo networks, they have a distinct spatial location. They have an identity. And so you can you can basically use that as a criterion for connectivity. Are they close together, or are they farther apart? And then you can threshold that distance, and you can calculate out like a network topology, given like maybe you say that you want a really high threshold for distance, so you want to look at all the cells that are connected. If your criterion is like a very small circle around each cell, and there are a number of connections there, and usually the cells that are close together will cluster into modules, and those that don't, won't. And then usually those correspond to these sublineages in the C. elegans lineage tree. And so this is useful because in biology, the cells are signaling one another. They're sending out chemical signals from each cell, and they're communicating. It's called paracrine signaling. And so a lot of times that's important for development. It's more important for those sort of regulative embryos that we talked about earlier. But in C. elegans, you still have that kind of signaling. And so these kind of networks will tell you maybe something about that kind of signaling. It could tell you about what positions they need to be in to differentiate or something like that. So, building a network from like microscopy data, especially if you're using like a graph neural network approach where, there are different methodologies for creating the graph neural network, message passing, or some of these other methods. I think you said, I don't know what you're thinking about for sort of a criterion. I mean, we could even work out a biologically specific criterion for like building a graph neural network and just building the embedding from the data. Because we have like with the microscopy images, see this was taken from like cell tracking where you have like positions that are averaged. If we took like a bunch of microscopy images and took each stack and said, we're like a series of images over time, and said, maybe that if the cell is moving or it's migrating to a final position, we could actually calculate graph embeddings that sort of capture that movement or the spatial location relative to cell divisions. We still haven't worked that out very well with this method. But like, with just taking it from microscopy data and building embeddings, you can kind of get these graph topologies that are, maybe change with time, and they change their shape or something. And so, that's kind of where this might be useful, and kind of giving that description of what cells sort of cluster together. Do they lead to the formation of tissues? Do they change the shape of the network? Which is these are all questions that even in network science, people haven't really gotten a handle on. Like, if you have a network with this shape, like it's like 3 modules, that are kind of form a triangle, what does that mean? We don't know that. But these are kinds of things you could do with these kind of embryo networks. But I like, I think we need to kind of say we could probably implement something similar to this. But of course, with graph neural networks, it gives us the ability to generate these embeddings and use them to, to sort of say something about the embryo instead of just like cells and segmenting cells. So we get this extra layer of structure on top of that.
Mm-hmm. Yeah. Actually, yes, I agree with you. Actually, I think, like if we look at this paper, just like what you said, maybe we don't. If we sometimes we explore some kind of topology, but maybe it would be very difficult to say like to clearly understand what those topology means and how those topology works. and how those topology would affect other modules or other functions. But sometimes, like for machine learning and deep learning models, this topology, we can try to like input this detected topology into the deep learning models and see if this input is helpful for deep learning models to do some downstream tasks, such as the cell tracking. And if the performance, if finally performance say, yes, it helps, then we can see that this topology is helpful. Although although it's still very difficult to explain how this topology help. Yeah.
Yeah, I think it's sort of an open question. We don't know like why it would be important for a certain topology over another. We just, I mean, in network science, a lot of the state of the art is like, well, actually, people have done a lot of things with some things with topological data analysis, but it's very early days. Like if I showed you, like they have these things called hairball networks, and it's sort of like this, except there's no structure at all. It's just kind of like this mass of connections, and people have identified things like that, but and clearly you can like visually identify things like modules or like clusters, right? But knowing what those mean and kind of getting at the structure of those and their relationship, that's sort of, they've tried to do that with community detection, where they kind of try to detect these subgroups or these communities within the network hairball or within the network topology. But that's like a very hard computational problem. You have to do like, you have to basically bootstrap where you have to do jackknifing or bootstrapping or some technique like that to which is not like compared to deep learning, it's not that computationally difficult. But you have these networks that have like a lot of nodes and a lot of connections, a lot of links. So you're dealing with a lot of it's I don't know, maybe there's a shortcut using deep learning. I don't think anyone's looked at that, but interpreting that topology, I agree, is a very important thing to sort of get our hands around. But really isn't a lot of state of the art that we can draw from. We just have to kind of come up with something.
Yeah. All right. Okay. Let me go ahead and because thing is I don't want to take up too much time actually. Yeah. And actually in this paper the connectivity is I think at the end of my presentation I would introduce the persistent homology, which is to detect multiple dimensions of topology, and zero dimension topology in the context of persistent homology is exactly the connected components, which is to analyze or to detect the connectivity. So actually in this paper, I would like to say the connectivity introduced in this paper is exactly the zero-dimensional topological signature detected by the persistent homology. Yes, but so uh, uh, apart from the connectivity, we have some other papers, and these papers they are also introduced in our group meeting, in the DevoWorm group meeting before. For example, in this paper, the author showed that I think for some organisms or for some multicellular organizations of diverse systems, they observe some kind of like global rotation of those collectivity cell migration. Yeah.
Yeah.
So this is one kind of like high dimensional topological signature. In the context of persistent homology, we would like to say this kind of rotation, it is a circle, or we can say it is a set of circles. And circles in the context of persistent homology is the one-dimensional topological signature. Actually, yes.
Yeah.
And if you go to this is another paper, and it also introduced in the group meeting before. And I think this paper is wanted to say that the collective cell migration is a very important process, and the substrate or interface of this process are typically curved. Like, for example, if you look at this picture, the substrates, there are this like a semi-sphere maybe, and these substrates they're curved. And actually, in persistent homology, we say if we just look at the homology theory, homology tour, this theory is not able to detect, is not able to differentiate two spaces which have different curvatures actually. But the persistent homology based on these point clouds, there are some papers show that persistent homology is able to detect the curvature, the difference of the curvature. So it is helpful to differentiate these spaces if these spaces have different curvatures. So actually sphere or things related to the balls or the voice or the gravities or something like this, they are higher dimensional topological signatures such as the 2 or 3 dimensional. Yes. And this is another paper, the growing topology of the C. elegans connectum. And this paper is interesting because it is actually using the persistent homology to analyze the topology along the development of C. elegans connectum. And if you look at, there are some results, and I want to show one plot. And if you look at this figure, many persistent cavities emerge in the developing neuron connectum of C. elegans. You can see that there are some names like barcode, Bailey number, or and each density. And actually, barcode and Bailey numbers, they are names from the homology theory, especially the persistent homology. And if I won't like introduce the definitions of these names, but I would say the Bailey numbers show that if Bailey numbers is very big, that means there are lots of high dimensional cycles, such as the cycle, or such as the how to say, the voice, or such as the connected components, which are a lot of these topological signatures in this space. And you can see that the Bailey number is very big, like 60, 80, or 100. That means there are lots of high dimensional signatures in this space. And if you look at this barcode, you will see there are lots of lines which are very long. If these lines are long, that means there are lots of signatures, topological signature, they born at a very early stage and they die at a very late stage. So like these signature, these cycles, they live for a very long period. Maybe it is very difficult to understand what does it mean for a signature they can live for a long period or for a long time. But in the context of persistent homology, that means there are some important topological signatures. Yeah. So I think I won't introduce the persistent homology today because we've taken a very long time. But what I want to say is, we can see that there are lots of topological signatures during the cell development process, and so then, but actually in what I want to say is actually in those previous works of cell tracking tasks, including those classical works and including those deep learning works, those methods are essentially based on the visual features of cells. Like they think these two cells, they are visually similar to each other. Like their visual traits, they are very similar to each other, so they connect these two cells. So these are the essential ideas behind those previous works. I'm not sure my statement is correct because I still need to refer to a lot of other papers. But for now, for me, in my opinion, it seems that there are few works considering the topological information within the cell development process to conduct the cell tracking task. So I would like to bring the persistent homology, this tool, to analyze the topological information within the cell development process and use this information to enhance the cell connection model or cell tracking model, such as the graph neural network that I introduced before. And actually, there's a very simple demo pipeline for this this idea. First of all, you have persistent homology, and let's just call it pH for simplicity. And you have pH, and assuming that pH is a model, and you input the points cloud you have into the pH, and pH generates some results, some outputs, which is exactly the persistent diagrams. And then you use some methods to vectorize the persistent diagrams. So that's the persistent diagrams are converted into some embeddings or some vectors, right? So actually, in this process, there are many problems. For example, this conversion or this mapping is not it is not differentiable. That means it cannot be like automatically differentiable by those PyTorch or TensorFlow or some other frameworks. But it is minor problems, and we would solve this in the future. But for now, after that, the persistent diagrams are converted into the embeddings. That means you have an embedding, which is the topological summary of your data sets. Like, for example, if your data set, there is a global rotation in the cell development process, then this embedding can summarize this global rotation, because the global rotation is a set of circles, is a set of one-dimensional topological features, which can be detected by the persistent homology. Yeah, that's my point. So that means you use persistent homology to detect those global topology. So after that, when you have the topological embedding, apart from this topological embedding, you have another embedding, which is the visual embedding generated by cell segmentation model. Maybe we want to say, okay, we don't need this visual embedding, but actually because we need to do the cell segmentation stage, and we will have a cell segmentation model, like some CM models, and if we have these models, we don't need to remove the outputs of these models, which is the visual embeddings. Yeah, maybe I didn't make myself clear here, but yeah. So you know what I mean? Yeah. So when you have topological embeddings and you have visual embeddings, you have a lot of ways to combine them, and you can use something like concatenation. You can use some MLP models to combine them, and there are lots of ways in the deep learning scenarios. So it is not very challenging. But after that, after you combine these two embeddings into a single embedding, that means this embedding, it has topological information and it has the visual information of cells. Then you want to input like this embedding into a cell connection model, such as a graph neural network, and you can take the same process of the graph neural network paper that I introduced before to do the link prediction, to connect cells. So that's all. That's a very simple demo pipeline. And actually, during this whole process, we have a lot of problems to solve. For example, So the differentiability is the first problem that I introduced before. And another problem is you may still remember that the topological embedding is a topological summary of the whole data set. But what we want to do is to connect cells. So that means we are doing work on the cell level. We need the information of each single cell. But so that means we have a requirements. That means we want to map the topological summary of the whole data sets into the cell level topological information. Yeah. So this is another challenge. Yeah. So actually for now there are lots of other challenges, but I won't like introduce all of them here. So that's my idea for how to use TDA for the cell tracking. And for some other stuff for me, because I'm a machine learning guy, I'm a deep learning guy, I know more about the deep learning and less about the biology. So if we wanna do a research and prepare a paper, and we wanna target a conference or journal, for me, just for me, I know one conference, the ICAR, the ICLEAR, which will happen. Not happen. I mean, the deadline for the submission for this conference, in general, the late September or the early October. Maybe I remember the conference will happen in Austria in next year. I'm not sure. So maybe it is very interesting. So in my plan, because actually this is because actually I'm now pursuing my MPhil degree. Maybe you don't know what is the MPhil. It is the Master of Research in Hong Kong. Yes, and it is one of my research topic during my my study. Yeah, so in my plan of timeline, because it is today it is 19 June, and maybe in June, July, and August and September is 3.5 months. I would like to focus on this project, and like if we can conduct any works on this idea. Yeah, for now.
Yeah, that would be great.
That's all.
Yeah, that would be great. I'd be happy to supervise it and make sure that we can get something out by probably the fall. Be great. Himanshu had a couple questions in the chat. One was, does the single embedding that is generated work on multiple downstream tasks, or are you planning to focus on a single task that can be then branched out?
Yeah, actually, this very cool question. Actually, for now, what I'm focusing on is only the cell tracking task. But I think because the multicellular biology is a very big field, there can be many other tasks. I think maybe many other downstream tasks we can focus on. So I think for now we can have a demo. Like we have a demo. If our method can work on cell tracking, we can consider more tasks. And if you have any idea about other tasks you want to try, we can consider them, because I think our method, this persistent homology and the graph neural network, I don't think these methods are restricted to any downstream task. It is not limited to the cell tracking, I think. So maybe we can consider more tasks, I think. Yeah.
Yeah. It sounds like he's pretty excited. Yeah, thank you. Thank you for the. And so Himanshu said, I kind of understood what Jiahang said. We'll have to revisit this. So yeah.
If you have a question, you can ask me. Maybe in a Slack channel, or maybe something else. Yeah.
Well, thank you, Jiahang, for that explanation. It looks really interesting, and we'll revisit this in upcoming meetings and work it out.
Yeah, of course. Yeah, thank you.
Thank you. So it looks like Sushmanth joined us after Jiahang was started his presentation. So Sushmanth, I know you presented on Friday. Did you want to add anything or?
Yeah, I want to add some things. I just missed out my train, so I couldn't go back.
Oh yeah.
So this is I have worked on lately this week after my devs meeting. When I had meeting with my dev, I was not able to iterate through the create a batch of bounding boxes. So Mayuk also told to drop the idea of fine tuning SAM, but I tried a little bit after that meeting. Somehow made out to take a batch of boxes at same time. Here we can see I was able to iterate over train loader, data loader. I took a batch size of 8, and these are the image dimensions. This is the mask dimension, and this is the box dimension. There are 8 boxes of 159,4 of first box. First box contains of 159,4. So it is working pretty well. So I was going through the SAM architecture image encoder. I was almost at the end of this code. I just need to write the training loop and train it on the loss function. Right now, I was working on this loss function, which I caught my interest. This is a special function. Generally, it is used only for segmentation things. Right now, we are giving the model the bounding box, and it tries to segment the image in that bounding box, and then that which have been predicted will be compared with our original ground truth. According to that, this loss function works. And I have read so many articles around this. So they mentioned like this is the only best method. This is the only best loss function we can use to get less loss in the model. So the code is almost ready. I just need to write the training loop. I was using Adam Optimizer. Everything was fine. I just going through the image encoder part. We are not changing any image encoder part, but I was changing a little bit in the decoder part. As it is a 90 million parameter model, so I was not touching any prompt encoder or vision encoder and image encoder. I was just changing the weights of the last layer of decoder layer, which segments the whole thing. So yeah, that's what my update is. Maybe I will start working on this training loop also from tomorrow. I have some resources to work it on, and and another thing. Now this is another thing I have got. This is a resource. There is some tutorial on how to fine tune SAM. So here they were segmenting they are giving annotations for each file, which I don't have. So I was just normally implementing without these annotations, right? Yeah, I was trying to replicate this code and implement in a nice way. After this segment anything model is done, then I will move on to the DevoNet model and start working on it. Actually, I wrote a little bit code of dataset, how to convert a 3D TIFF file into normal ND array. I will start working on the other things also. So yeah, that's what my update is. Maybe from tomorrow I'll start working on the training of the model also.
Well, that's great. Thank you for the update. So it sounds like you got what you needed from the meeting with Mayuk on this weekend. And
he helped. Actually, he told to drop the idea of fine tuning SAM.
Yeah.
He told to the model architecture is kind of bit different and not able to. We can't fine tune it. That is the reason he told to drop the idea.
Yeah.
And he suggested me to work on the Deep Lab V3 model. But I thought of giving it a try. Then when I implemented this thing, I was able to get a data loader, which is iterable, by using collate function. Actually, the Jiahang Li is what you have mentioned in the channel, DevoLearn channel. It helped me. I was going through some other people codes, and yeah, I was able to write it. Yeah, maybe. I will start working on the training loop also. Maybe I will train this weekend the model. After that, after we getting the losses and the segmentation loss of the model, I will start working on the DevoNet model. Yeah, that's what my update is. And I was thinking to convert those. Right now, we are converting the membrane segmenter position as a centroid, but I was thinking to take the cell segmenter position, cell's position as a centroid. That's what I was thinking about, because I don't know why we took a centroid of a cell. And Mayuk also told me to start working on the DevoLearn. There are so many deprecated libraries. He told to it's not working properly.
Yeah.
He showed me in the last meeting the tests are failing. The library is almost deprecated. We can't use it. So he told to work on it. Day before yesterday, I have tried to run the tests on this on my forked repo. This is what happened. I was able to pass the normal test cases for cell segmenter, lineage, cell membrane segmenter, and the GAN model, but the tests are not passing for the lineage population model. There are some deprecated libraries, mainly scikit-learn library, which is not passing the test cases. So I was going through them. Maybe, yeah, I'll show it here. I tried to update it, but I couldn't get time due to this traveling and all. Yeah, just let me show it once. Yeah, here it is mentioned like scikit-learn library is almost deprecated. Where could I say?
Yeah.
Here it's some, yeah, scikit-learn, yeah. It's mentioned like no module named sklearn, because sklearn has been changed to another name called scikit-learn. Yeah, that's what the problem it is. I will update this requirements.txt, and I was trying to integrate depend-about, which I have told last week, into the DevoLearn, because whenever the libraries are deprecated, it automatically changes the deprecated libraries and updates into the new ones. That's what we had in the meeting.
Okay. Yeah, that sounds great. Then the question you had about centroids, or like how to define a centroid, or
we need to find the centroid of a cell, right? I mean,
right.
where the cell is located. But right now, whatever our model is there, it is trying to get centroids from the membrane. Why we are taking the centroid of a membrane?
Well, we can't necessarily define the nucleus. So like the when you have like say an image that like a fluorescent image where you have like this plate area inside the cell, that's usually the fluorescent marker, but that's not necessarily in the nucleus. It could be in the membrane as well. So we want to get the centroid of the membrane and find like the center point. Now that being said, it doesn't necessarily have to be based on the membrane. It could be based on like, some other criterion. If you think there's some other way to do it, that's better.
I was thinking to first segment the cells. After that, upon the time frame, when the segmented cell, I will try to find out the location of the cell. That we can use as centroid rather than a membrane centroid. That's what I was thinking. I mean, if you think this as a 3D space, here the cell is located. So that's located cell centroid position in the XYZ plane. It will be mentioned. It is located in the 2, 3, 4 point, or rather than a centroid in a membrane, we can represent a segmented cell position according to the time frame.
Yeah.
That's what my thinking is. I will try to implement it. I will let you know by next week. I just need to train this model. Still, there are so many bugs coming out. I tried to train today only, but train it today only. The image architecture is so different. I don't want to like see how this goes on, but I will try this week completely on this model. If I couldn't get anywhere, I will start working on the DevoNet. Actually, it's taking a lot of time rather than I expected. The image architecture was like bit different compared to any other models. Here they are segmenting cells according to the prompt we give. not according to the ground truths of the masks generally we have. So yeah, I will try to implement this week as soon as possible and update you by next week. After that, I'll start working on the demo.
Okay, and yeah, I have to request your extension, so let me know, and yeah, I'll just do that. And,
Okay, yeah. Let me know. Maybe. Yeah, I still didn't get how much time I need. I will plan it out.
what was that? Oh, plan.
So finding a bounding box of a cell, then do a computation on that. Yeah, right now we have the ground truths of the masks, right, for every cell image. So from the ground truth mask, I was extracting the bounding box, and that bounding box was given to Sam prompt as a prompt. Then it will segment the, already it was pre-trained, right? So it will segment the required cell in that bounding box, and that is taken as to compare with the loss with original ground truth mask. That's how it is. Let me show it to you. Yeah, okay. This is the architecture actually.
Okay.
This image is fed to an image encoder. So here is the mask, but we need to give the model, not mask. Generally, when we train any model, we generally give the ground truth mask, and we'll try to train it. But here we can see we can give a prompt as a point or a box, bounding box, and a text also. But this version of thing is not released yet. They only given the version which we can segment through box or points. So for the prompt encoder, we are giving the bounding box, which will be in the form of xmax, ymax, xmin, and ymin of the box, and that forms a 4D box around it, and that is given to a prompt encoder, and the image is given to image encoder. Then we will get a mask at our image decoder, what model is predicting. Then we will compare it with our original mask, and then we will try to find it out whether we are, and we'll try to calculate the loss of the model. Yeah, that's how the model works. This is the architecture of the model. Yeah.
Right.
Yeah.
Yeah, and then that's the performance. So you could basically take like a point. Anything is like a prompt. Like you could have the bounding box, a spatial point, or some text.
Yeah.
Okay.
Yeah. We generally give a bounding box or a point, just a position on the object itself that generates the position. I mean, that gives that when we give it to the model, it will try to predict that whatever or the surrounded objects are there. It will try to segment it, and then we will compare with the ground truth what we have. Then the losses are calculated. That's what.
Yeah. Yeah, that looks pretty good. Thank you.
Yeah,
thank you for the update. So, do we have any other updates before we go? Jesse, did you want to say anything or anything?
Hey, not really. Just it's good to see everyone here again. I know I haven't been around a lot, and seeing the progress and seeing Jiahang and seeing everybody from past and present and future. Not a lot of updates. Just I'm going to try to be doing a little bit more in the space, and really cool to see the developments that have been made here. So maybe more in the next few weeks, but it's good to see everybody this time. And that's all. Take care.
All right. All right. Thanks for attending. Have a good week. Thank you.